So good morning, everyone. You're very welcome. My name is Anne-Marie O'Dowd, and I'm chair of Cystinosis Network Europe, which is an umbrella organisation for patient organisations all over Europe. I'm also the mother of a young adult with cystinosis, which is my most important job. First of all, I want to tell you about the translation. So anyone using translation, there's a QR code on your table, and it will open the translation app. And now I realise I probably should speak a little bit slower if there's people using translation. So my job this morning is to thank the people involved in this conference. A huge thank you to Cystinosis Foundation UK for hosting the conference on behalf of Cystinosis Network Europe in this lovely city of Manchester. Thank you to the organising committee who have put in enormous work to get us all here today. Every detail has been considered, and so much thought has gone into every aspect. Thank you to our sponsors. It's worked. And they have helped make this conference possible. We're very lucky in the cystinosis community to have so many scientific and clinical experts working in the field. It is the reason that we are continually increasing our knowledge, and it's the reason we have a treatment, unlike about 90% of other rare diseases. And it's why we have such hope for the future, for new treatments coming down the tracks and new knowledge. You'll hear all about that in the coming days, and you'll be able to talk to people. And you have that unique opportunity here this weekend to engage with those experts, and I urge you to make the most of it. Talk to them. If you've been to the conference before, welcome back. You'll be renewing connections and friendships, and you'll know how powerful that is. If you're a first-timer wearing the pink badge, we're very glad you've made it. We hope that you learn some new information, but mostly we hope you'll make your own connections and friendships. You might feel a bit overwhelmed at times, and that's normal, so take care of yourself. But know that you're among people that understand where you really don't have to explain here. So while there might be the occasional tear, there will definitely be laughter, and there'll even be dancing. And so I will let who's the chair of the Cystinosis Foundation UK and also the chair of the organisation committee, tell you all about the dancing. But first, I am going to introduce you to Dr. Graham Lipkin. Graham is a consultant nephrologist, a kidney doctor in the University of Birmingham, Queen Elizabeth Hospital. He has a long-standing interest in adult cystinosis, and he's involved in development of the national clinics, designation clinics, and he's engaged in the pro-drug program. Thank you.
Thank you very much, Anne-Marie. And as chair of the scientific committee, I'd just like to add my warm welcome to you all to this biennial Cystinosis Network Europe meeting here in Manchester. We're really grateful that you've attended, but you'll be rewarded by a superb program over the next few days, something to look forward to. Yesterday, we held a scientific meeting focused entirely upon issues related to cystinosis and had a really far-reaching and successful day as a precursor for today. But what we wanted to do is to bring that knowledge into this meeting today. So you'll hear after, just around lunchtime, the highlights by the scientists and clinicians, experts in that area. And we hope for a better treatments ahead, better organization of service delivery, and there's been an enormous amount of work in various countries, as well as the development of new guidelines, which you will hear. This conference is almost unique, bringing together patients and their carers, scientists, expert clinicians, and is used really to be an opportunity for you to influence those people and for those people, the scientists and clinicians, to be able to feed back to you where we're going. So I will just end by thanking my scientific committee. That's Elena Levchenko, Konrad Vase, David Game, Will, Denise, Ruth Davis, and Neil Sugden. And I wish you a wonderful couple of days. I think it will be well worth the visit. Thank you.
Morning, everybody. Those of you who were here last night at the welcome will be fed up with my voice by now, but those of you who weren't, welcome everybody. And it's my huge pleasure and privilege as chair of Cystinosis Foundation UK and the organising committee for this conference to welcome you all here today. So many welcomes. It's 10 years, apparently, since the last big international conference here in Manchester, which was organised by my predecessor, Roy, who's sitting there, who is relaxing now because it took years off his life, apparently. But yeah, so back in 2014, I'd never heard of cystinosis, and I know there are people here who are in the same boat who didn't know, but there are also people who were here 10 years ago who've come back and met people for the first time since that time. So it's remarkable. I think it's absolutely brilliant. And that was happening last night. So the reason I know about cystinosis, my granddaughter Ellie was diagnosed in 2016, which obviously brought cystinosis into our family's life. And I suppose as a newcomer, if you are new here, you're going to hear a lot of information. You're going to go through a lot of emotional turmoil. It's going to lift you up. It's going to drop you down. Do take care of yourselves when you're here. Do think about, do I need to take a rest if I want to walk out of this? It's getting too much. Do feel that you can do it. It's not formal that you have to sit there and listen to absolutely everything. You know, you choose, you make those decisions. Yeah, your brain and your emotions are going to need a rest. So our wonderful organising committee started planning probably last March for this conference. Lots of WhatsApps, lots of Zoom calls, and it wasn't till October that we met here, well, in the bar here, as a group, and we suddenly realised what a difference that made, just actually meeting in person together. So hence, it was so much better together, and hence that reason for the conference. So thank you everybody for coming. Patients, individuals with cystinosis, carers, grandparents. You've got a huge contribution to make talking to each other and listening to each other. Colleagues from the medical profession, Graham, and the others, you know, thank you for looking after our loved ones. You know, it makes a huge difference. Welcome to our talented researchers and dedicated researchers who are always looking for new ways, new treatments, new ways to make our loved ones better and prove that those treatments. And thanks also to our colleagues from pharmaceutical industries who actually make those treatments a reality. The whole conference, I think, as Graham said, it's unique, that people can come together, share ideas, share experiences, so that we can be better together. Our organising committee set me a little challenge. They like doing that. So because we have people, it's an international conference, I've got a list of 15 countries here, which they may not all now be represented because we have had some people who couldn't make it, but here we go anyway. I am going to shout out the name of a country. If you just wave or do whatever you want, you can dance on the table if you wish, and I'll try and say welcome in your language, so we'll see how this goes. Careful, Franz, careful. So we'll start with, this is alphabetical order. I was a librarian, so everything is always in alphabetical order. So Belgium. Welcome. France. Bienvenue. Germany. Willkommen. Ireland. Falscher. Netherlands. Welcome. Norway. Velkommen. Portugal. Benvindo. I don't think Saudi Arabia came, but I'm going to say it anyway if they haven't, because I learnt it. It's Marhaban Bikum. South Africa. Welcome. Spain. Bienvenidos. Switzerland. Willkommen. Turkey. Hoş geldiniz. United Kingdom, hi. USA, howdy y'all. Okay, just a few housekeeping things. I have been told there is no fire alarm or practice intended today. If anything does go off, we have to get out of the hotel and meet by the big flagpoles at the entrance. Canada. Oh, heck. Hiya. Yeah, the way things are going to work today is there will be a set of presentations, and at the end of those, an opportunity to ask questions. If you don't feel brave enough to talk through the mic that will be going round, there are cards on the tables, piece of paper, there will be there. So if you want to write a question, and then we will get somebody to read it out for you. Is that all, Alex? That's it? I think that's probably it. So we've got a quick quiz, set of questions for you on something called Menti. So we should have a QR code coming up. So you'll need your phone. Okay, so if you scan that, you should get, what will it say on their screen? Okay. Yeah, so it should say, welcome to the CNE conference. Yeah? Okay. Oh, hang on, we've got some still looking. How many? 39 so far. Come on, we can up that. 40. 50. 55. Still waiting for Canada. Yeah. 65. This is good. 72. 74. Okay. I think, yeah, there's one phone left up. I think I've got it. 79? 80? Okay, so a first question should come up now, which is a test one. So you just need to press one of those options. Okay. Okay. So it's hovering at the moment about 8.3, is that? 5.3 out of 10. Haven't used it before. 22, what's that, 2.2? 22% have. And 14% yes, but not much. And somebody said, I can't remember, but that wasn't me. Okay, next question. So who's in the audience, and you select the option that most aligns with why you are here. Oh, wow. So we've got a lot of family members here, 39, 42, 43, 15, 16 who have cystinosis, 11 healthcare, 6 charity, 4 researchers, 7 pharma. Yeah, so we're up to 53 family members, right? Next one, where have you traveled from? Is Canada on there? Oh, no. North America. So in the lead at the moment is the UK, and Europe's nearly taken over. North America's heading that way. Oh, it's neck and neck between Europe and UK. Europe are ahead. The UK has suddenly left Europe again. Yeah. Yep. And is this your first time at a cystinosis conference? So 33 yes, 67 no, 35 yes, 69. So that's a third, 2/3 roughly, isn't it? Yeah. Oh, golly, this is a toughie. Could you summarize the impact of cystinosis on your life in one word? And if so, what would that be? There's going to be a gap while people think about this. Here we go. Heartbreaking, challenging, you're in, difficult, an adventurous challenge. Oh my goodness. Difficult. Caregiving, love, hope. And it's getting too small for my eyes. So the biggest thing at the moment is challenging and difficult, life-changing. Manageable. Yeah. Can we save that to share that? Yeah, that would be really good. Yeah, so we'll get like a word cloud. And last question, how are you hoping to feel after this conference? Informed, hopeful, energized, grateful, inspired, educated, connected, like that, more aware. Yeah, informed, hopeful, and educated are winning at the moment, and connected. That's really good. Yeah, that's excellent. That's what we hope from the conference too. So thank you very much. And I'm going to hand over now to our next speakers, I hope. Excellent. Thanks, Alex. Thank you.
So good morning and welcome. My name is Francesco Emma. I'm chairing this session, and I have the task to introduce Elena Levchenko. So who is here at a cystinosis conference for the first time? Okay, then for you and only for you, Elena Levchenko is a professor of pediatrics. She is also the chief of pediatric nephrology in Amsterdam. And she has devoted her research life to cystinosis. She has published over the years few papers on cystinosis, more than 90. And I think most importantly, she has supervised clinicians and researchers that are now working on cystinosis. So she really deserves a lot of credit for this. She works closely with the Patients Association. She's funded by the Cystinosis Group from the Netherlands and Belgium and by Cystinosis Ireland. Before being in Amsterdam, she was working in Leuven. Before that, she was working in Nijmegen. Before that, she was in Belgium. Before that, she was in Russia. So I can hear you asking yourself, oh, is she very old or is she very smart? The correct answer is number 2. So please, Elena.
Good morning, everybody, and thank you, Francesco, for this very kind introduction. I was a little bit scared what he's going to say about me, but it was okay. So it's really my great pleasure to be again at this meeting, and this meeting is really unique. It feels like a family, and I am very fortunate to be a member of this family already for quite some years. And it was a great privilege to be a part of the organizing committee. So today, I'm going to tell you something about cystinosis research. And because there are some new families, I will start from the very beginning, even from the time when I was not yet born. And on this slide, you see our enemy in cystinosis, cysteine. It's an amino acid. And this amino acid was actually the first amino acid which was described in the world. And it was described by a chemist who analyzed their stone found in the bladder of the patient. And this stone is still kept in the museum in London, and it appeared to be cysteine. And this stone was found in a patient with a different disease than cystinosis. It's called cystinuria. And these patients form kidney stones, and patients with cystinosis don't form kidney stones, but it allowed to discover this amino acid. So the first description of the disease is attributed to this guy. His name is Emil Abderhalden. He was a chemist. At that time, mostly chemistry made progress. And he made an autopsy in a small child who died from a failure to thrive. And in the tissue of this child, they found this amino acid, cysteine. And he, for the first time, described the disease. He called it cysteine diathese. That was a name he gave to the disease. So then in the 20s and the 30s, there were several physicians who were describing the kidney disease in cystinosis. First, it was Dr. Lignac, and then cystinosis was called the disease of Lignac disease. And then there were De Togni, Debré, and Fanconi who were describing the kidney dysfunction, the proximal tubular dysfunction with a lot of urine production, loss of phosphate, rickets, glucose, and then the disease was called De Togni, Debré, Fanconi syndrome. And finally, we still call the kidney presentation in cystinosis renal Fanconi syndrome. But at that moment, we really didn't know, or people didn't know, what was the real biochemical basis of cystinosis. They found cysteine crystals, but they even didn't know whether these crystals were inside of the cells or outside of the cells. And finally, it was Jerry Schneider, and he's really the godfather of cystinosis. Jerry Schneider, for the first time, showed that cysteine crystals are inside of the cells. He could measure it. He developed the technique to measure cysteine inside of the cells. And also he published this, for example, very nice image showing the bone marrow aspirate in cystinosis patient, showing the cysteine crystals inside of the cells. And this was very important. So we understood the biochemical basis of cystinosis, being the accumulation of cysteine crystals. Another very important person was Giustini. Giustini, while Jerry Schneider passed away a few years ago, and many of us knew him, he was a very inspiring person who really inspired a lot of people in cystinosis field and treated also a lot of patients in the United States. Giustini is still active. He participated actually in the development of very recent guidelines in cystinosis. And at that time, it's also many years ago, he worked together with Dr. Oshima and Dr. Schneider in the lab, and they tested different compounds to decrease cystine accumulation in the cells of cystinosis. And they tested different drugs, and finally, cysteamine, the drug that you all know very well, was very efficient to deplete cystinotic cells from cystine, and you see the more they gave, the lower cystine concentration was in the cells. And they published again a very nice paper, and then they were able to immediately go from the lab to the patient. No animal models, no ethical committees. They just could inject cysteamine first intravenously into the patient and measure cystine content in blood cells, and then they developed the oral preparation, and then that's how cysteamine entered the field. It was a short way, and they just did it. I think if these studies would take place now, I think that Dr. Tini would probably still be submitting papers and providing their safety, and all kind of officers would look at this file, and it will not be very easy. But at that time, things were much easier than nowadays, and this is how cysteamine came into the clinical practice. And this is a very busy cartoon. You see, I don't know whether this is working. Yes. So here you see the lysosome. Actually, it is very important that cystine is not only inside of the cells, but inside of the lysosomes in the cells. Cystine accumulates. Cysteamine, which is shown here, can enter the lysosomes, and it can break cystine in two other molecules that can exit the lysosomes via the different transporters, and that's how the drug works. It breaks cystine inside of the lysosomes, and it depletes lysosomal cystine accumulation. Another person who is very important in the cystinosis field is Dr. Gall. Many of you who have been to the previous conferences know Dr. Gall. He worked all his life in NIH, and he followed a lot of cystinosis patients, and he did wonderful, really very important studies for the field. And this is one of the very important studies that demonstrated for the first time that cysteamine works in cystinosis patients to protect their kidney function. And you can see here on this graph, so these are the years, and this is the creatinine clearance of the patients. You see that when patients were not treated with cysteamine, all of them developed kidney failure before the age of 10 years, while patients who were well treated with cysteamine had preserved kidney function. Again, that was a very important study, and based on that, the whole world started to use cysteamine in cystinotic patients. Dr. Gall also published papers in cystinosis patients, and he looked at the extrarenal complications, and here you can see different of these complications such as diabetes, myopathy, pulmonary dysfunction, also death, and he had demonstrated that patients who were on cysteamine for many years were protected, or at least these complications could be delayed. So again, that was a very important publication. And then there was, for some years, quite quiet on the cystinosis research field. And it's very interesting. We knew the molecular basis, we had the treatment for the disease, but we still didn't know the gene. And it's only in 1998, it's a consortium which was guided by Corinne Antignac and Paris. They were able to clone cystinosis gene on the chromosome 17. You see the human chromosomes, and here you see chromosome 17, and there where cystinosis gene is located. So they provided the genetic basis of cystinosis, the gene where the mutations are found, and now we know more than 160 mutations in the cystinosis patients. Okay, they also have demonstrated that the protein is located on the lysosomal membrane, and that they demonstrated that exports cystine together with protons out of their lysosomes. And so the picture was complete. We had the gene, we had the protein, we had the mechanism, how it works. And that was the end of the 20th century. So we were quite satisfied. We could make the diagnosis of cystinosis very reliably, could measure cystine in blood cells. We could confirm the diagnosis by analyzing the gene. We could treat the patients by administering oral cysteamine, but also by administering the topical eye drops. So, and that was something that we could be happy about. We could postpone kidney failure with about 5 to 8 years, and we could postpone extrarenal complications. Well, but not really good enough. I think that's not where patients were searching for, and also not us as physicians. So I think patients really wanted more, patients wanted cure, patients wanted more quality of life, and it was also something that I think is very important for the research agenda for this century we are living now. So what are we trying to achieve now? I think we are really moving from postponing disease complications to the cure. And this is very, very important. There is still a lot of work to do, but we are really moving in this direction. I think we are talking now about preventing disease development rather than treating the disease. And we have seen results showing that a very early diagnosis can really prevent the disease development. The researchers are also working on the patient-friendly cysteamine formulations. You know that the cysteamine is a difficult drug to take. It has a lot of side effects, but there are perspectives in these directions as well. I think we are still getting more and more insights in understanding the disease mechanisms. And this results in finding new treatments other than cysteamine, and these treatments will probably also change the life of cystinosis patients. We also learn how to better organize the care of cystinosis patients, how to monitor patients, how to support the families. And this is especially true for adult patients who are more spread around the country. And altogether, it should result in the better quality of life and actually normal life expectancy for cystinosis families and patients. Just looking at the time, how many minutes do I have left? Okay, so we can still talk further. And so I just would like to highlight a few very recent research developments, and you will hear more about these research developments. Obviously, I cannot talk about everything. Yesterday, we had an excellent day with the researchers. I think everybody was really inspired and excited, and there is a lot in the pipeline for the future of our patients and families. So I think one of the very exciting things you will hear about today is an update on the hematopoietic stem cell transplantation of patients' own stem cells, which are corrected with a healthy cystinosis gene. That was a phase 1, phase 2 clinical trial performed by Dr. Stephanie Cherki. And now this trial is finished. 6 patients are transplanted, and there are a lot of promising results, and I think you will hear about these results, which is very, very interesting. Another very important development is the possibility of the neonatal screening in cystinosis. And this is Katharina Hoffenfellner, who did this pioneering work. First of all, we know that the cystinosis is a disease having the latent period. Patients are born without symptoms. And this gives us a window of opportunity to diagnose the disease and to treat it very, very early. And therefore, cystinosis fulfills the criteria, the international criteria for the diseases that can be selected for the neonatal screening. It is a severe disease, it is treatable disease, and we have this latent period where we can do something before the patient develops symptoms. And Katharina was the first time to demonstrate the feasibility to diagnose cystinosis during the neonatal period by analyzing the DNA isolated from the dry blood spots, which we all use for the neonatal screening. What was very important in this regard that she also published 6 patients who were treated by cysteamine before the age of 2 months, so very, very early, before renal Fanconi syndrome was developed. And they have shown that 4 out of the 6 patients treated so early did not develop renal Fanconi syndrome. They did not need any supplements for the renal Fanconi syndrome. And this is extremely, extremely important. This means that the early administration of cysteamine can prevent the development of renal Fanconi syndrome, at least in some patients. Another paper was published by Konrad Weiss. He compared the renal course in siblings of patients with cystinosis. The older sibling was diagnosed when the symptoms were already present, and the younger sibling, shown here in blue, was diagnosed because the disease was already known in the family at the neonatal period. And you can see that these siblings, which were treated starting from the neonatal age, none of them developed kidney failure until the age of 20 years. Again, demonstrating that when you treat cystinosis patients very, very early, you can prevent the development of kidney disease. So I think another very, very exciting finding that we were discussing yesterday, and you will also hear an update on this study today, is the development of cysteamine prodrug. So this work was started by Rose Anderson in the University of Sunderland. She was a very talented pharmacologist, and many of you who were at these meetings before have met her. She was always among the patients explaining her work. And unfortunately, she passed away a few years ago, very, very sad, at a very young age. Fortunately, Professor Herbert Newell took over this project, and this project is progressing very, very well. So what they thought, they decided to improve the existing cysteamine by binding the molecule of cysteamine with 3 other compounds, which is thioester, shown here in green, which guards the sulfur of cysteamine and protects the stomach and gut. So this gives less gastrointestinal side effects. It has an ester, which improves the reabsorption of the drug. So the drug goes much faster than cysteamine to the bloodstream. And it also has a glutamate, which targets cysteamine for being released on the cell surface. So it's more targeted drug than the cysteamine that many of the patients are taking now. So very interesting compound. There is a lot of work already done in cells, in the animal models. They had to go through all the regulation to put the drug on the market. And I was very, very happy to hear that the phase 1 clinical trial is really going to start this year. And I think this is extremely, extremely exciting. I have real big expectations from this compound, from this new drug. So during the last years, we were working on developing models of cystinosis because you need models to test new treatments, to test new drugs. And now we have a lot of interesting models, cell models, which can be cultured in 2D. We have tubuloids, so kind of the kidney tubules that were developed to test the drugs. We have organoids, which are kind of the mini kidneys, which can be also cultured in the culture dish. We have animal models, the mouse model, the rat model, which recapitulate the kidney and the other phenotypes of the disease. We have the fish model. So we have really tools to test new treatments. And these studies showed us that our favorite drug, cysteamine, next to correcting lysosomal cystine accumulation, acts also on the other mechanisms in cystinosis. It's a very complex slide, but I just give you some flavors of these complex processes in the cystinosis cells. So cysteamine can also decrease the production of reactive oxygen species, which are damaging the cells. It can also decrease cell death. Fine, this is very, very important mechanism in cystinosis cells. It does something on the mitochondrial function. Mitochondria are organelles which are producing energy. But we note that when renal Fanconi syndrome is established, cysteamine is unable to correct renal Fanconi syndrome. And this is very unfortunate, and this is what we are struggling. And a lot of people are struggling with this renal Fanconi syndrome. So what can we do about it? So there is a lot of research going in this direction. And a very recent study was published by the group of Francesco Erma, actually by his younger colleague, Francesco Bellomo. And they applied a diet. The diet, which is called the ketogenic diet. Many of you have heard about this diet. In this diet, the calories are mainly provided as fat, and the carbohydrates are really very much restricted. The protein content in this diet is more or less normal. This diet is known for ages. It is used to treat epilepsy seizures, and also is tried in many other diseases like cancer or some inflammatory diseases. And what they have done, they gave this diet to their animal models of cystinosis, to the mouse and to the rat. And what they found was very, very interesting. The animals that were treated with this diet, and these animals are indicated in red here, you can see that when this diet was administered at the age of 3 months, at this age, this mouse don't have renal Fanconi syndrome. The development of renal Fanconi syndrome could be prevented. You see, this mouse had the normal urine production, no glucose in urine, no proteins in urine, so this mouse was just healthy, which was not the case for the mouse treated with a standard diet. But what was even more exciting than when they gave this diet to the mouse that already had renal Fanconi syndrome starting from the age of 6 months, the symptoms of renal Fanconi syndrome could be reversed. So urine production went down, glucose excretion went down, proteins went down. So there was something happening that could reverse renal Fanconi syndrome in these animals. And this is very, very interesting. Please don't think that everybody starting from now has to be eating this ketogenic diet. It can also have side effects. It should be all tested because it's not just something you can do starting from now, but I think it gives us a promise and it might help us. How can we treat renal Fanconi syndrome in the patient? And maybe there are even some drugs that can mimic this diet without being obliged to follow this diet, which is quite difficult to follow. So the last thing I would like to mention, because I think it's also really very much exciting, is the use of mTOR inhibitors in cystinosis. This is also a very complex slide. You see again the lysosome. Yeah. As I can find my pointer. So here you can see the lysosome, and here you can see the mTOR complex 1 on the lysosomal membrane. You see it's sitting very close to the cystinosin, and this is a very important nutrient sensor. So when cell has nutrient, mTOR is active, it's on. And it stimulates protein synthesis, it stimulates cell growth. So very important sensor. But when there are no nutrients, mTOR will go out, it will be located in the cytosol, not on the lysosomal membrane. And at that moment, the cell starts what we call self-eating, because mTOR, when it's active, it inhibits autophagy. When there are no nutrients, the cell will start self-eating, it will start degrade own proteins, own nutrients. And in a way, it's a healthy process for the cell, because the cell cleans itself from the old proteins, old organs. So this is also an important process. So several groups have shown that in patients with cystinosis, mTOR is a little bit crazy, or maybe very much crazy. It is always on. So it's not sensing well enough. It stays there on the lysosomal membrane, and it starts really working. And this is not a healthy process. And so what was done by Jennifer Hollywood, and she will also present this data today. She used a drug which is already on the market, mTOR inhibitor, everolimus. We know this drug is an immunosuppressive drug. It's used in patients after kidney transplantation. It's used in some cancers and some other diseases. And they gave this drug to the rat model of cystinosis that they developed in their laboratory. And again, what they could see, and especially the combination of mTOR with cysteamine, could not only deplete cystine accumulation. Actually, this depletion was much higher than cysteamine alone. It could also decrease urine output, decrease excretion of the proteins, again, having a positive effect on the renal Fanconi syndrome. And these are really first treatments that have impact on the renal Fanconi syndrome. So very interesting development, and I hope that it will be also coming to their clinical trials, and we will see how it will work, but it gives us a lot of promise. So I think I would like to stop here, and I think we are living in a very, very interesting time period. We have the unique opportunity to work together as a community. We have a wonderful participation of the patients, of the families. We have the foundations. We have a very dedicated group of clinicians, scientists, researchers. We have also industry who is dedicated to cystinosis. So we are really in the opportunity to change the future of the cystinosis patients. And I think the future is really going in the right direction. So here you see the natural history of cystinosis patients before the 70s. Most of the patients died of kidney failure because a kidney transplantation was not yet available for children. Then in the next years, the future of the cystinosis patients has improved. We could postpone kidney failure, we could postpone extrarenal complications. But now, we see the whole spectrum. We can diagnose cystinosis at the neonatal age, and we see the growing generation of all the cystinosis patients. Our oldest cystinosis patients in the Netherlands, she's older than me, and I think it's for many of you, it's very, very old. And this lady is still in a good condition, and I think there are a lot of patients who are passing 60, cystinosis patient passing 60, and then I think it gives really promise to all of us. So I think the future is bright. And let's further work together on the future of cystinosis patients, and I would like to stop here and to thank you very much for your kind attention.
Well, thank you very much, Elena. This was a great presentation. I think it was an outstanding opening for this meeting. And now it's my great pleasure to present to you Stephen Jenkins. So Stephen comes from Salt Lake City. He has trained as a specialist in internal medicine at University of Utah, and he is currently an associate professor at the University of Utah, and he works as a hospital specialist and a thrombosis specialist. And he is also the father of two sons that have cystinosis. And for that, he actually serves in the Cystinosis Research Foundation Board of Trustees and in the Cystinosis Research Foundation Scientific Review Board. So you must be asking that he is really very young and that he's very smart. And actually the correct answer is both. So Stephen, what are you?
All right. Thank you, Dr. Emma, for that introduction. I don't know if I'm very smart, but I feel young, and I hope I can be young for a little while longer. We traveled here, arrived on Tuesday, and brought our children with us, including a 3-year-old toddler, and she makes me feel not very young. We haven't had much sleep over the last few days, but we'll see if we can get through this. So, thank you, Dr. Levchenko, for that overview. I think that was the best overview I think I've heard of the history of cystinosis research and kind of putting it all together. What I'm going to be talking about is very, very basic understanding of kind of how cystinosis works. And this is kind of how we've figured out how to explain it to our own children. And I think that even though, most of us are not going to be trained geneticists or nephrologists, we can all become experts in cystinosis by trying to understand it at this level. And I think it helps us, understand what treatments are coming down the pipeline and how we can be better advocates for ourselves and our children. So we call this the building blocks talk. Sometimes we call it the Lego talk. So I'm up here today because I have 2 children with cystinosis. This is a picture of my older son, Samuel. He's now 15 years old. But this is a picture of him when he was basically right after his first birthday. And in a story that's very familiar to all of you, I'm sure, around the age of 7 months, he stopped growing, he stopped eating normally, he was throwing up all the time, he was getting dehydrated, and was very thirsty. If you put a bottle of water in front of him, he went completely nuts. And we were pretty worried, and we started taking him to a lot of doctors, trying to figure out what was going on with him. And around maybe the age of 10 months, we were at a family reunion. And my grandmother, she saw Samuel and she said, "Wow, he's acting just like your cousin Amy and Emily when they were babies." And when I heard her say that, I kind of froze. So I knew that my cousins, Amy and Emily, had a rare genetic disease. I knew it was called cystinosis, but I didn't know that much about it. I knew that they had kidney transplants. I remember my parents telling me that they probably wouldn't live, very long into adulthood. And so I was pretty terrified when my grandma said that. This is a picture of my cousin Emily. She passed away a couple years ago at the age of 40. And she was a very, very funny, entertaining person, and I have lots of good memories hanging out with her at family events. She was just a few years older than me. And so I knew that, she'd had a lot of health problems, but I didn't really know much about cystinosis at that point. So we got Sam to a nephrologist. We got him tested for cystinosis, and the results came back positive. And we got him started on treatment. And this is a picture of him when he got his gastrostomy tube just a couple months later, so that we could help get him the nutrition that he needed, get him the medicines that he needed. And you can see, he's trying to put a smile on, but it was a pretty tough situation. I think we were in the hospital for about a week longer than expected, just because the doctors couldn't figure out how to keep his electrolytes balanced and got him a little bit fluid overloaded, but we made it through that. And here's him just a couple years later, and you can see he's got his gastrostomy tube there, and we were connected to that feeding tube all the time, right? And so I know many of you have probably dealt with this, and I thought Samuel is never going to eat food. At this point, he was only drinking water, and then all of his nutrition was going through that tube. And so I really did wonder at that point if Sam would ever be able to eat like a normal kid. Around the time that Sam was about 2 years old, we thought, things were starting to get a little bit easier. It was never easy managing cystinosis, but we felt like we had the routine down. We thought, man, we'd really like Samuel to have a sibling one day, and thought, well, we don't want him to get too old. Let's see if we can have another kid. And we understood that there was a chance that our next child could also have cystinosis, but we thought, it's a 75% chance that he'll be healthy, or he or she will be healthy. So we rolled the dice and had another kid. And this is my other son, Lars. I don't know how old he is here, but we got him tested for cystinosis right after he was born, and he also came back positive. And that was pretty disappointing. At the time, we were pretty sad that Lars was also going to have to go through some of these things, but fortunately, because we knew he had cystinosis at a very young age, we were able to get him on treatment right away, get him on the cysteamine, and it's made a huge difference for him. He doesn't have the Fanconi syndrome like Samuel, and he's really only taking the one medication, the cysteamine, at this point. And so This is just a couple of years ago. Lars is on the left side, Sam is on the right side, and even though, it's really sad that they both have cystinosis, we're very grateful that they have each other, and they've been really great support to each other. And cystinosis is a really challenging disease, but we're really grateful for them. So Sam did start eating food eventually, and eventually he was able to get that gastrostomy tube out when he was eating food and taking his medicines. And so this is one of his friends, Henry Sturgis, who lives in Idaho, and they were both pretty proud to show off their scars from getting that G tube out. We've been going to conferences in the United States, ever since Sam was about 2 or 3 years old, and it's been a real, huge support to us to meet other families who understand cystinosis, to have these other children with cystinosis in our lives. And last year we thought, after we went to a conference, man, it would be really great if we could spend a little bit more time with these kids. The conferences are never long enough, and so we decided, well, let's do our own cystinosis summer camp. And so we invited some of their friends from the conferences to come to Utah. And so we had 8 kids with cystinosis come last summer to our place, and we hosted them for just a few days, and it was way harder than I thought it would be. It turns out 17-year-olds, even though they're teenagers, they think they're adults. But we had a lot of fun with them. We took them down the Provo River, which is a very mild river, not really like any rapids, but we had a lot of fun rafting down that river with them. We took them to some hot springs, and it was kind of fun to watch them immediately get in the water together, and they started synchronized swimming together. Like, they just really immediately bonded with each other and had a great time. So we are going to try to do a summer camp this year again as well. And so, if you have teenagers and you feel comfortable sending them to the United States, we'd be happy to host them. But maybe next year, if you're interested in a summer camp, let us know. So that's a little bit about our story, but now I want to talk more about cystinosis and really those basic concepts. So to really understand cystinosis, you have to have a basic understanding of cellular biology. So everyone kind of learns about the cell in school, but just as a reminder, the human body is made up of 40 trillion cells, and each cell has different compartments in it. So you'll see here, this large compartment is the nucleus, and that's the most important place probably in the cell. That's the command center where all the DNA is stored, the genetic material. But you have other organelles as well, like the Golgi apparatus and ribosomes and something called a lysosome. They all have different important functions. So like we were saying, the nucleus is where you store your genetic material, and that's your DNA here, which is represented by these letters. And the DNA is a double helix. It's wound up into these structures called chromosomes. So all of your DNA is stored in chromosomes. And here's a karyotype that shows your chromosomes. Everyone has 23 pairs of chromosomes. You get half from your mom and half from your dad. You'll see here the 23rd are your X and Y's. And so when we think about what DNA is actually for, it's written instructions for the cell on how to build proteins, right? So you have DNA, these letters, and you have special machinery that's going to open up the DNA and transcribe those letters into a script called RNA. And then that RNA gets fed through a different machinery called a ribosome, and that's going to generate the protein, which is kind of an abstract concept. The way we use to explain this to our kids is with Legos, right? So the DNA is your Legos. Sorry, I got like some kind of automatic play going here. My apologies. So you have your instructions are your DNA. You have your master builder, which is this gentleman here, which looks like Emmett from the Lego movie. He's your ribosome. And then your actual building blocks are your amino acids, like cysteine. Okay, and then your finished product that you're going to build with the Legos is your protein. So those Lego instructions are the DNA. So what if you have, part of the instructions are missing? That would be your mutation there. So when this guy comes to build the Lego set, he doesn't have the instructions anymore. So now your race car is missing that spoiler that was part of the race car. So that's a mutated protein now. And there's different types of mutations that you can have. You might just have a point mutation where one letter is wrong. You might have an extra letter get inserted into the code, and then that kind of shifts everything down. Or you might be missing a letter in the code, and that also will shift things. Or you might have entire sections that are missing from the DNA code, like deletions. And so, like Dr. Levchenko was saying, we've discovered that cystinosis mutations are located on the 17th chromosome. That was discovered by Dr. Corinne Antignac back in '98. And they found where that gene was on that 17th chromosome. And since then, more than 100 different mutations have been identified. The most common is called that 57kb deletion, 57kb standing for 57,000 base pairs. So 57,000 letters have been deleted from that DNA code. That's the most common mutation in folks of Northern European descent, but there's also lots of other mutations you can have. So when we had our children's DNA sequenced, we were surprised to learn that neither of them have, or that they don't have that 57kb, but they have two other mutations that can cause the same phenotype. So we say that cystinosis is an autosomal recessive disorder, which means you need two copies of the mutated gene in order to have the disease. And so if you get a copy of a gene from your mom and a chromosome from your dad, if there's no mutation on those, then you're going to have an unaffected child. But you'll see here that mom also has this dark blue. So you can also get a normal copy from mom and a mutated copy from dad, and that child will be a carrier, right? They'll have a copy of the mutation, but it won't be expressed. You can have a copy from your dad and a normal from your mom. Oops, my apologies. So you can have a mutated copy from mom and a normal copy from dad, and that child will be a carrier. And then it really requires that you have a mutated copy from mom and dad for the child to have cystinosis. So you can see here that 25% chance each time you have a child that they'll have cystinosis, but 75% chance that they'll either be a carrier or unaffected. So this is a cartoon from a New England Journal article about cystinosis showing the cystinosis protein here in the membrane of a lysosome. And it's showing all the different types of mutations that you can have. But I think the important thing to note is that it is here in a lysosome. And so talking about lysosomes, these organelles in the cell are really the recycling center for this cell. This is where all the trash goes to get recycled and broken down. This is where old proteins go. If the cell, is responsible for, fighting off bacteria or viruses, those things are going to get digested in the lysosome. And so this is a terrible cartoon of a lysosome, but basically if you have an old protein, like this race car here that comes into the lysosome to be recycled. You have enzymes like Lloyd the Green Ninja here who are going to break it down into different amino acids. And then each of those amino acids can leave the lysosome through a different transporter protein. So in the case of cystinosis, or cystinosin, is a transporter protein that is responsible for getting cysteine out of the lysosomes, right? So cysteine here is represented by this blue Lego. And in the case of cystinosis, that transporter protein is defective. And so that cysteine is going to build up in that lysosome. And so because that's affecting every single cell in the body, it's going to affect all the different organs in the body. And so we often think of cystinosis as primarily affecting the kidneys, but we know it affects all of the cells in the body. And that's why there's so many different systemic problems with cystinosis. So we know that it affects the eyes, but it also affects endocrine organs like your thyroid gland, the sex organs, the pancreas, and that can cause different problems. We know it affects the bones and the muscles as well. And so really we should think of cystinosis as being a cellular disease that affects every single cell in the body. And so Dr. Levchenko talked about the discovery of cysteamine and how that works. But basically, cysteamine is a molecule that can enter into the lysosome. It's going to bind to these cysteine amino acids and form a new molecule that can actually exit the lysosome through a different transporter protein. And so that's how cysteamine works. It basically goes in there and just helps deplete the cysteine by getting it out through a different transporter protein. Cysteamine is a miracle drug, I think. We're very fortunate that we have cysteamine. There's many lysosome storage disorders and other genetic disorders that have no treatments available. And so I think even though cysteamine is a really tough drug to take, we are very lucky that we have it. But it's not perfect. It's not a cure. We know that it delays complications of cystinosis. It does not, entirely prevent them forever. And so I think we all look forward to the future when we could have other treatments available. And one of those potential treatments would be a gene therapy. And so Dr. Levchenko talked about Dr. Sherky's work, where she's worked on lentiviral gene therapy with autologous stem cell transplants, where basically you take a person with cystinosis and you remove some of their hematopoietic stem cells. You use a virus to insert a copy of the corrected cystinosin gene into those stem cells, and then you give them their own stem cells back to them. And because it's their own stem cells, you're not going to have issues with the immune system, either rejecting them or those cells attacking that person like you get with graft versus host disease. You give those cells back, and that could potentially be a treatment for cystinosis. And when I first heard about that, I thought, well, geez, that doesn't really make sense. Cystinosis is a disease that affects every cell in the body. It's not just affecting blood cells. So why would, giving someone basically a bone marrow transplant, a blood cell transplant, why would that help someone with cystinosis? It's interesting to learn, though, that some of those blood cells that you're replacing are called monocytes. Those cells turn into macrophages, and macrophages are these large cells that literally, macrophage means big eater. It's the cells that go around and they one of their jobs is to clean up messes, right? So if you have an infection and your immune system fights off a virus or bacteria, and there's a big mess left over after that infection, one of the jobs of the macrophage is to go in and clean that up. But they're doing other things too. But what we've learned is that these macrophages are loaded up with lysosomes, because one of their jobs is to clean up messes, and that's what lysosomes do, right? Those are the recycling centers. So these macrophages are traveling around the body to clean up messes. They're packed with these lysosomes. So what Dr. Sherky has found is that these macrophages actually create tunneling nanotubules, is what she calls them, these little projections where they can actually insert these projections into other cells, and they can share these corrected lysosomes. So basically these are healthy lysosomes that these macrophages are sharing with these injured cells, or like cells with cystinosis. And then the bad lysosomes, or the affected lysosomes, are then swapped out by these macrophages. So this is a pretty incredible concept, I think. You have macrophages that have been corrected by gene therapy that are going around the body and basically sharing healthy lysosomes with the other cells. And one way I think of that is basically these macrophages are like Amazon delivery trucks that are just loaded up with lysosomes, and they're there to deliver them throughout the body. So I think that is all the slides that I have. But just wanted to thank you all for your attention. Thank you for this opportunity to come and be with you here in the UK. I think one thing that's remarkable to me is that, no matter where you go in the world, as soon as you meet other people with cystinosis who are affected by cystinosis, you immediately have a bond with them. You immediately feel like you're part of a family with them. And I've felt that here in the United Kingdom as I've talked with you guys. When I got here, We realized that we had forgotten some of Samuel's medications. We didn't have his phosphorus supplement that I somehow forgot to pack that. And we're going to be here for a little while. And so I started to panic because you don't want to miss your phosphorus supplements too many times in a row. You can get low pretty quick. And so I'm immediately getting on, Facebook and asking people, what do you guys use for phosphorus in the United Kingdom? And I emailed Will Newman and said, what do you guys use for phosphorus here? And I ran to a pharmacy and luckily they had some phosphorus supplements that they were willing to sell to me without a prescription. I guess you don't need a script here for them, so that was convenient. And so Sam's been choking down this phosphorus supplement here. It's the fizzy tablets. We don't use those in Utah, but apparently they're pretty common here. And he's now started calling them the devil's drink because it makes him belch a lot and it's not very pleasant to drink. But it was very, very grateful to Will Newman and others who I talked to from the UK who said, oh yeah, that's what we use too, and that should work. So I think we're all sorted there. But it's been a pleasure to be here so far, and I look forward to talking with the rest of you. So thank you.
Thank you both. I think this was a wonderful opening for this meeting. Coffee break is in 13 minutes, so we have time for 13 minutes of questions to Elena and Stefan. Who wants to start? Oh, here we go. Please.
Never the shy one. Hi there. Thank you for those wonderful presentations. They took me back to the 20th century when our kids were diagnosed. A very similar story to you, Stephen. Having a child and then deciding that you had a 75% chance of having a second child who may be okay, but wasn't. So brought some tears to my eyes just listening to your story. But luckily, through all the research that Lena's been talking about, they are thriving. They're in their 20s. They're sat at the back somewhere, and dealing with life. So, thank you for those presentations. My question was, when our eldest, not the eldest, the second-born child, so we have got a third child who's older without cystinosis, but when Isha was diagnosed in '98, we saw Cystagon had come onto the market, and it was the wonder drug. And in the last 20 years, the hope for kids has got that much brighter. Do you think that speed of the medicines and the speed of countering the disease will get back quicker and quicker to find the ultimate cure, which may be gene therapy? Or do you think actually it's going to take a lot longer because it's quite a complicated cure?
All right.
Your answer.
I think it's a great question, and I think Dr. Levchenko, I think, showed really nicely how, there's been like periods of time where there's been kind of a lot of accelerated research and then kind of some lulls. And I feel like we are in a period of accelerated research right now where there's lots of different people working on cystinosis from many different angles. People looking at different prodrugs, people looking at gene therapy, people looking at autophagy. There's all these different, mechanisms of cystinosis that we're just barely understanding, and all of those open up different potential targets for treatment. And so I think we are definitely kind of in a renaissance right now of lots of potential treatments. So I'm hopeful. It gives me a lot of hope that I think, like I talked about, my cousin Emily, she had Cystagon, and now we have Procysbi, and hopefully we're going to have the CF10 in the near future. And gene therapy it gives us a lot of hope.
Yeah, I fully agree. And I think what is happening now in cystinosis is also happening in other disease fields. And the great thing is that we are learning from each other. So the technology that are being developed in cystinosis can be applied for the other disease fields, and vice versa. Other technologies can be applied for cystinosis. And what we have learned, and was a very nice presentation of Alexandra Luciani yesterday, that we can also apply artificial intelligence for things, because you need sometimes to analyze enormous amount of data or information where human brain might be even too small for or not smart enough for. And then we can use the artificial intelligence. And actually, mTOR was one of their targets which was popping up using this artificial intelligence tool. So I think the progress is really now accelerated, and probably it will be faster and faster. So I think for many diseases, including cystinosis, we will be able to find cure and have much better quality of life.
Thank you. And maybe would you like to make a comment on what it takes to bring a drug to the patients, that we can no longer do what Dr. Turney and Dr. Gall did, that they just took the power and just gave it to the patients. Maybe it's worth making a comment on that.
Well, this is something that is also part of the modern time. I think there are questions of privacy and the questions of ethics. Also, because of this huge opportunity to collect information, to share information, this is an opposite side of the medal. We should be very cautious by doing that, because there we come to the issues of privacy and protection. And so, in my opinion, the balance is now really a little bit skewed towards overprotection and overregulation. You cannot imagine the amount of papers you need to fill in and the amount of bureaucrats looking at these papers and checking each letter and comma and point. So this is, but people are aware of this, and there are also publications suggesting how to facilitate this. Maybe also there should be some artificial intelligence instead of human beings controlling these letters. So I hope that this part of the medical research which accelerate as well, but we are not yet there, and this is something which really slowing down research, in my opinion, very much at this moment. But we need these people. We can really don't do it as Dr. Tini did. It was very good, and many research discoveries at that time were done like this. But we're in a different time period, and we should be very cautious, I think.
Yeah, I think cysteamine is such a toxic drug. I don't know if it would ever clear regulatory hurdles anymore, but I feel like as a parent, I'm always very anxious for these treatments to come to my kids now, right? And we've been coming to meetings now, for I guess 13 years now. So I've been hearing about things coming down the pipeline for that long, and it's difficult. You have to be patient. But I think, I've learned from Dr. Emma from some of his research, he's screened big drug libraries to look for potential treatments for cystinosis, and you find these very promising potential drugs that are already approved, and you could just go pick up some of them at the over the counter of the pharmacy, and you're like, whoa, we should just go start those right now. But when you actually study them, like in a rat model with cystinosis, and you find, oh, actually that drug has lots of potential problems and side effects. And so you don't want to just go put that in your kid right away. You really do want to go through all of the proper steps to study it and make sure that it's safe. So even though it's difficult to be patient and to wait, I think it's important that we go through the proper process and protocols to make sure it's safe for our kids.
Well, thank you, Stephen. This is what I wanted to get to. It's important to realize that there are safety checks which are absolutely needed before a drug is brought to you, and not going through this procedure can be very dangerous, and this is why it takes a little bit longer. 4 more minutes. Who wants to ask another question? There is one question.
Good morning. I'm not really. Good morning, everyone. This is my first time coming here, and I'm not really talkative, but I just want to know from this gentleman, how did you take the Mickey button off your child, and what was your journey like, and what kind of food and the medication helped? And thank you very much for coming and inviting us here, because thank you so much.
Oh, thank you. We're glad you're here. like I said, I didn't ever think Sam would get his tube out. It felt like it would be there forever. And really it was just, you know, he started eating food maybe when he was 5, or maybe 4 or 5, mostly because his little brother was eating food. And I think he felt competitive maybe with him, or maybe just curious, like, oh, maybe I should put something in my mouth too. It took a long time though. And I don't know that there's any special tricks. I think it takes just time and, you know, sometimes some occupational therapy and things to work on that. Eventually he was eating, but then we were still using that gastrostomy tube for his medications. And there was one medication in particular that he would not take by mouth because it tasted so bad. And I don't remember how old he was. Maybe he was about 12 or 13 when he finally decided, I'm going to take that medication because I don't want to have the tube anymore. And so really it didn't matter how much we pushed him. He had to decide that he was ready. We waited several months after that to make sure that there was not going to be any backsliding. And you know, when he showed that he could do it, we got it out. Many people just remove them and they'll seal off on their own. We did have surgery to remove it just so they could, you know, make a nice smaller scar, but that was all pretty straightforward. So I think really it just takes some time and patience, and every child is a little different how fast they want to go.
All right. Well, time for one last question, if there is one in the room. Here it is.
So first, thank you very much. That was the best movie I've ever seen to explain cystinosis to normal people. It was really, really nice. And I was laughing about your Lego persons. It's just a remark I want to make. When speaking about medication, about new medication and about over-the-counter medication, what you just said, like something like this, all this costs a lot of money. And some people can't afford even the medication, over the counter. They don't have the money to pay for it. And so we have to keep in mind to keep all treatment payable for everybody. And there are lots of very, very, very expensive medications that are not offered in a lot of countries. Mm-hmm. So we still have, I think we have the duty to work, not only work on new medication, but work on payable medication using the old, somewhat older things as well, to have everybody treated all over the world in the next few years, because they can't wait anymore.
Hear, hear.
We can only agree on that. I think this is a very important remark, and I think, we should keep in mind.
I think the applause speaks for the rightness of this remark, and I think it's the best way to close this session. Thank you very much, and thank you, Elena. Thank you, Stephen, for these wonderful talks.